In this video, we will understand about some of the assays that can be used to detect UPR detection. In our previous videos, we have understand what is UPR response and how it is generated, and what is plasmid, and how it is used to express a, express a DNA of our interest. So, UPR is known as unfolded protein response. Just for a revision, it is usually occurred when there are unfolded proteins due to any reason increased in an endoplasmic reticulum. So UPR leads to three major signals. One is PERK, one is IRE1 alpha, another one is ATF4 translation. However, in this video, we will discuss about how to measure PERK activity sensor, and how to measure PERK activity and IRE1 activity through the sensors that has been developed. Though there are many other assays available, however, we will focus upon one of the most recent essay that has been published. It is published in 2018 in Assay and Drug Journal. I don't remember the exact name of the journal. The exact name of the journal is Assay and Drug Development Technologies Journal. So, as we have discussed in plasmid, what we do is we inject the DNA of our the DNA, not the DNA, we can say that sequence of the protein of our interest in the plasmid, and then transfect them into the cell lines for the expression, for its expression. So this diagram here, it shows the sequence or the how the sequence is made, so that we can measure the desired response, that is PERK activity and IRE1 activity. In case of PERK activity, when there is no ER stress, usually the translation occurred in the open reading frame 1, and when the translation occurs in open reading frame 1 and it goes on, and when the UPR response observed, one of the function of the PERK activity is to reduce the translation of the protein that are not essential at that point of time for cell survival. So for that, what actually happens is that instead of translating from open reading frame 1, it starts translating from open reading frame 3. Yeah, and previous sensors, what has been developed is that they they have created a modified fluorescence protein in the open reading frame 1 or frame 2, so that when the ER stress occurred, the translation of those proteins get reduced and the fluorescence signals get reduced. However, in this sensor, what they have done is they have introduced a fluorescence modified or recombinant fluorescence protein in the after open reading frame 3. And what they have included three major things. One is the HA tag, and I will tell you what's the purpose of HA tag. Another is M scarlet protein. It gives a fluorescence signals. It gives a red signal, red fluorescence. And another one is NLS sequence, that is nucleotide nucleotide localization sequence. Nucleotide localization sequence. This HA tag is produced so that they can test their whether the sensor is working or not with the help of like Western blot techniques. I have not so far explained about Western blot, but why this HA, I will explain about Western blot in one of the next videos. And but for now, let us understand that this HA tag, the anti-HA tag, that is anti-HA antibodies, that is antibodies that can detect HA tag, is are been easily available and are more specific. So if one need to see whether this code of interest is expressed or not, they can easily extract the proteins and go and do the Western blot against HA tag, and from this they can understand whether this kind of proteins, whether this code has been translated or not. If this is translated, then they can have a idea, then they can have much more confidence that their per activity sensor is working. However, it is possible that without ER stress it can be translated due to some other biological cross talks or cross talks or some other reasons. So in order to cross confirm that ER stress is generated, and because of this and because of ER stress this translation of this sequence happens, what people have done is they have also did a Western blot on one of the proteins that has been confirmed to be generated only when the ER stress is ER stress occur, that is the CHOP protein. So how they have confirmed that per activity sensor is working? They first induce a ER stress with the help of two compounds. One is DMSO, or another one is its name is tyrosine. Spelling might be wrong, or I can write it in the short form. Or tyrosine. When this is introduced, what it will does is it will reduce the transfer of Ca2 into the endoplasmic reticulum. So that will lead to that will unstabilize the hemostasis in the endoplasmic reticulum, which result in UPR response, and so that UPR response will lead to translation of this CHOP protein. So in order to confirm whether the sensor is working, people have done the Western blot not only of this anti-HA tag to confirm this sequence is translated, but also of the CHOP proteins to confirm that it is also been translated or produced in the cells. So by doing Western blotting of these two things, they can confirm that per activity sensor is working. And for IRE1 the process is same, but just they have to make a different sequence that can sense whether IRE1 activity sensor is activated or not. So what IRE1 when IRE1 does when it is activated, it XPB1 usually is unspliced in normal condition. When ER stress is observed, what it does is, this is the intron. This intron is removed and then it is sliced. Intron is removed and sliced, and now this intron is in the base sequence for 410 to 633. So it is sliced in case of ER stress, and now these two get combined. This spliced form of XPB1 then result in the production of more ER related components and ATF6 also. So how they have modified is that they have first include the HA tag in the start of the XPB1, and then they have introduced the modified fluorescence protein, some part of it before the intron and some part of it after the intron. So when this intron gets removed, it can translate, this sequence get translated into a fluorescence protein. Not the fluorescence protein, this sequence get translated sorry, what I have said is not right so far. So in case of no ER stress, this remains unspliced. XPV1U remains unspliced and it does not it is now in a form that does not get translated, so it will not produce subsequent reaction. However, in the case of ER stress, this intron gets spliced and now the XPV1 splice can be translated and produce downstream signaling that result in reduction of unnecessary or reduction of non-important reduction of translation of non-important proteins, and at the same time controlling the amount of controlling the amount of proteins that are responsible or that are important for the cell survival. So how they have modified these sequences? First, they have included HA tag, and when they have not included the intron, they have excluded the intron part. So when the ER stress get activated, this the ER stress get activated. Oh no, they have included this intron part also in this thing. Otherwise, it won't give the actual signal of IR even. So what they have done is they have included HR tag and then this intron part, and then here they have included green fluorescence proteins and then NLS tag, that is nucleotide localization sequence. So when the ER stress get activated, this intron get removed and this will get translated and it will complete it translated in till here. UAA, I have represent UAA at each of the sequence. UAA, as we have discussed it one of our video, that UAA represent the stop codon, so translation stop at this side. So in this case, when the ER stress get observed in the cell, this intron get removed and this sequence get translated, which result in green fluorescence protein. At the same time, this NLS also gets translated. So this proteins get now goes into the nucleotide. Why this NLS sequence is introduced? NLS sequence is introduced so that all of the signal get concentrated into nucleus and we will get a good contrast image. However, if this NLS sequence is not there, then the fluorescence proteins might move around in the cytosol and the signals might not be that contrast, might not be that strong. Sensitive signals might not be that strong. Instead, the signals could be like more of like blurry form, because some of the fluorescence proteins might be in cytosol, distribute around cytosol. However, nucleotide size is very much concise, and so the contrast that we get with when the fluorescence proteins are in the nucleus is much more better than without nucleotide. Then this fluorescence proteins is spread around cytosol. I guess this might provide you an brief overview how this UPR detection assay works. This is just one kind of assay. There are other assays also available in the field. Okay. There are other assays also available in the field about UPR detection. Thank you.